now I have the luxury and the privilege of introducing our moderator for today, Matt Bresnahan, who has been a fixture of First Look for some time now. And Matt, thank you so much for your support of the event. I'll be around if we need any help. And I think Matt's going to explain how the pitch presentations are going to run.
Great. Excellent. Yeah, thank you so much, Darren. And thank you for all your great work in getting today's activity scheduled and produced and a success that we're all looking forward to participating with. So as Darren mentioned, my name is Matt Bresnahan. I'm a partner at Wilson Sonsini, law firm focused on working with technology and life science companies, based here in Southern California, where I've spent the last 15 years at Wilson Sonsini working with emerging growth life science companies from all stages of growth, from formation, financing, patent strategy, which is my core background and skills, and through exit. So particularly happy to be moderating the life science track. Looking forward to hearing from a lot of great up and coming technologies that have some great chance at commercialization opportunities. So in terms of format, what we'll have is a series of 3-minute presentations from the groups, and then we will have that followed with a 9-minute Q&A from a great group of judges that we have. And during that process, all of you should feel free to ask any questions in the Q&A box, and we will read them to the best we can. We'll get through all of them, time permitting. So with that, what I'd like to do now is bring on our judges, a great group of judges, as I mentioned. So we have Alice Lee, Alice Chang, and Gladys Nunez. And what I'd like to do is allow each of them to introduce themselves, the firm that they represent, and anything else they'd like to share. So could we start with Alice Lee first?
Sure. Well, thank you for having me today. So I am a principal at Alexandria Venture Investments, which is Alexandria's strategic venture capital platform when we actively invest in seed and early stage. And also we're very active. I think 3 years running, we're the most active biopharma investor in terms of volume. Today I represent the Seattle region, but we're filling in for our colleague Hallie, and tomorrow you'll see Blake Stevens, who is another principal in our Research Triangle Park. And for those who are unfamiliar, I think in the previous talk they mentioned, Lily mentioned Alexandria, so we're a real estate company, and that's what most people know us as. But we are like a pioneer in this area where we lead and operate and develop life science and technology and ag tech campuses all around the country. And then in LA, we also have lab space. So if you're looking for, you could always reach out to any of us at Alexandria, and we'll figure out who for you to best talk to. Thank you.
Great. Thank you, Alice. Alice Chang?
Hi, everyone. I'm the other Alice, I guess. Upfront Ventures, we are based in Santa Monica. We are an early stage investor, so Series C, Series A, and we invest generally across industries. I focus on the healthcare life sciences. My background is in biomedical engineering, spent some time in consulting, and I would say we are location agnostic, but 40 to 50% of our portfolio is in Southern California, so that's Santa Barbara South.
Great. Thank you, Alice. Looking forward to hearing comments and questions. And Gladys Nunez?
Hi, everyone. Hopefully you can hear me. My name is Gladys Nunez, and I am a principal in the Amgen Ventures Group. We are the corporate venture group for Amgen. I think one of the sponsors that works closely with SoCal Innovation. So personally, I'm based in the Cambridge, Massachusetts site. We are located, my partner in ventures, Janice Nave, is based in San Francisco, but we cover all geographies, US and Europe. As noted, we invest in therapeutics, emerging platforms, and digital health. Personally, I'm a pharmacologist by training. I used to work previously in Amgen's business development organization. In the ventures group, I've been here about 6 years now. And I do run the diligence on new investment opportunities, including therapeutics, digital, and I am also in charge of Amgen's seed fund as well. So I look forward to seeing the companies and learning more. Thank you.
Great. Thank you, Gladys, and thank you, Alice and Alice, for all of you for joining us today and participating and serving as a judge and panelist. So with that, why don't we turn to introducing our first company in the therapeutics track, which is Alyra Therapeutics.
Well, I think we've had our first issue with doing this virtually right now. It appears that our speaker has dropped off the call somehow. So we're trying to get him back on. I would suggest perhaps we transition to the next person. Yes.
Yeah. Okay, sounds great. Thanks, Darren. So why don't we move to Xiretsa, and then we can follow up with Alyra, provided that they're able to come back on. So Xiretsa.
Hi, let me just get this set up here. Okay, I'm ready to share the screen, right? You have me set up here.
Yep, you should be able to.
Okay. Well, hi, everybody. My name's Robert Pastrozny. I am co-founder of Xiretsa, and I'd like to have you imagine something for a moment. Just let's say you're a parent, and you have a little boy, and he's outside, he's riding his bike, he's having a blast, but he falls down and he scrapes his knee, and this little scrape gets infected and he dies. And then let's say you maybe get a little paper cut that gets infected. You have a very good chance of dying too. See, this is what happens when antibiotics doesn't work anymore. And if antibiotics don't work, then we can kiss goodbye to surgery as well, because opening up the body, that's too risky. So, I mean, imagine how humanity's quality of life would be in this scenario. It's just mind-boggling. And yet, we're already here. This is happening. Right here in the US, every 15 minutes, a person dies from bacteria that's resistant to antibiotics. And it's increasing at such a high rate that this problem is considered the greatest public health threat in the world. But there's hope in the form of a new class of chemicals we've discovered called ACEs. ACEs are unlike any other antibiotic, so bacteria have no resistance to it. And in studies we've done on mice infected with untreatable superbugs, we've had a 100% cure rate. ACEs they're easy to make, they're easy to adjust so that we can treat a wide range of diseases. The first being NTM lung disease. Now, this is a $2.5 billion market and rising 5% of cases each year. And the existing drug is only 29% effective with absolutely horrendous side effects like deafness. And ACEs have the potential to cure 100% of these patients. Now, our drug revenue projections are over $1 billion peak sales year 5 on the market. So our team is uniquely suited to attack a problem such as this. On the top, we have Guy Bazan. He is one of the world's top material scientists. We have 4 PhDs, and we have entrepreneurs with proven track records, like successful tech exits of over $70 million. And Xiretsa has currently raised $70,000 with $1 million in non-dilutive funding. And we're now asking for $500K to do a proof of principle studies against NTM and also file international patents. And the $500K would be in a safe offering, which would convert to preferred stock as soon as we do an equity round. And so here's the thing. This is not just an investment. This is something that can potentially save your life in the future. It can save the lives of those you love, and it can also save the world from deadly infection. So I thank you very much.
Great. Thank you very much for your presentation, Patrick. Turn it over to our esteemed panelists of judges. Would anybody like to ask the first question?
Yeah, no, I mean, I think it's recognized that certainly, antibiotics have had a challenge in terms of getting support. You have big pharma kind of pulling away because as much as it's a medical need, sadly, there are considerations that pharma is making in terms of what is the ROI for this. But I recently came across, I think it was maybe a month or two ago, about the Action Fund that is like well over $1 billion. Is that something you've looked into? Because I think that might be more readily accessible than investment like venture capital dollars at this point. That's my guess anyway.
Yeah, well, that's a great point you bring up. I mean, they have $1 billion in this fund. Big Pharma has set up for that. And we've definitely looked into this. We're looking for other avenues too. And the thing about us is we're a little bit different in some of the materials that we have. And so it's definitely a challenge. But, it's one thing. Like at this point, all of the drugs in the development chain, I mean, nothing is kind of adequate to keep up with the pace of resistance. And so we're kind of positioning ourselves at the right time to really attack this. And at a certain point, a lot of things are going to change because they're going to have to. I mean, it's a big thing. And so, of course, there's challenges, but we're hopeful that our technology is so radically different that we'll be able to cut through some of the problems. And we also found a niche market here that is making money, and it's a good area to go in.
Well, just a follow-on comment. One of the companies I used to work with when I was back in Boston also got grants from CarbX. Is that something you've looked into as well?
Yeah, we're applying to CarbX. We have other grants that we're applying to, and so we're expecting STTI. We're expecting some of those grants to help us. One of the other founders too on this, Alex Moreland, he's a great chemist. He can answer any of the kind of chemistry. He's been applying to a lot of grants. He can answer that too. That comes up. But we're applying to those, and so we're looking everywhere because we need all the help we can get here.
Yeah, in the investment world, you see this kind of like strange, like infectious disease was like not a thing. And then now it's like the thing, right? So,
and the thing about
it's going to be like there's going to be a threshold. We're going to hit it, and then all of a sudden, like the things that you're doing is like it's going to be this FOMO, right? For a lot of
people. And the thing, and what you bring up about COVID, that's a good point too, because a lot of people don't know that a large percentage of those COVID deaths are actually bacterial resistant. It's a high number. I don't know if it's 30%. But the New York Times just did an article about this. And so I think it's bringing a lot of this to the forefront. I mean, we see now how just this pandemic has changed, the world essentially. And I think we're close to 900,000 deaths. Through resistance this year, we have over 700,000 deaths. But in a few decades, that's projected to be 10 million annual deaths at a cost of $100 trillion. So it's a big problem. It's going to get bigger, of course. We kind of wait till the end, but I do think COVID is bringing this up, like you say. So that's a good point.
So Robert, this is Gladys from Amgen Ventures. A couple of questions. I just want to delve into the science a little bit. Are the ACEs small molecules? What type of molecules are they? And then secondly, can you just expand upon the proof of concept studies that you're looking for the RAISE? Are you looking to kind of go against kind of current treatments with, I know that it's kind of a combination treatment. In that sense, what assays are you planning to run? So if you could just briefly describe that, I would appreciate it.
Yeah, thanks, Gladys. That's a great question. I'm going to let Alex, our chemist, answer that for you. He's another founder.
Sorry, I was trying to get my video on, but now I'm on. So the first question was, is it a small molecule? And it is. And I missed the second question.
Okay, no, I just wanted you to expand on the proof of concept studies that you're planning. So basically, what sort of assays are you running? What is the comparator assays and so on and so forth? Sure.
So we've done in vitro work with NTM and our ACEs, but we'd like to do an infection model in mice. And to get to that point, we want to improve our chemistry a little bit. this was kind of the foundation of some of the proposals that we've written recently as well. And we have done some in vivo work with our compounds, but it hasn't been in the space of NTM because we've also been exploring other indications. So as Robert said in the presentation, the ACEs were 100% effective against a few superbug infections, and those were MRSA and multi-drug resistant Klebsiella. So we want to kind of transfer that and look at a similar kind of mouse model with NTM specifically.
Okay, so essentially the proof of concept milestone will get you some in vivo data for NTM, correct?
Yeah, that's
okay,
right.
Just very briefly on the chemistry about what's the size of the molecules. I mean, you don't have to kind of, go down to the last decimal point, but just what's the range, please?
600 Daltons to maybe 1,400 Daltons.
Okay.
So
all right, thank you.
Big small molecules.
Yeah. Thanks.
Could you talk a little bit about how you chose NTM as your initial indication, and then how much work would need to be done to pivot into, I guess, like a new indication?
Would you like me to answer that, Robert?
Yeah, go ahead. You take it.
So we chose NTM for a couple reasons. First, our compounds are fairly effective against it, which is a difficult thing. So NTM is pretty similar to tuberculosis, and the new compounds that they've made for tuberculosis aren't even very effective against NTM. So there's a big challenge there in getting activity. Because we saw activity, we thought that'd be something good to explore. And then there's a few other issues with NTM that make it hard to treat. So they're hard to kill. So a lot of the compounds inhibit their growth, but don't actually kill the bacteria. So you still rely on the immune system to really clear the infection. And ACEs, on the other hand, are very rapidly bactericidal, so they kill the bacteria really quickly. And then the other important feature is kind of related to PK, and that's that the ACEs do not move easily from the lung to the blood. And so if we're going to treat these NTM in lung infections, we will have reduced systemic side effects relative to other compounds that they're currently using for NTM lung disease. Sorry, what was the second bit of that?
How much work needs to be done for you to address a different indication?
Right. So since we're so early, a lot of the parameters that we're trying to tune are going to be similar. So in every case, we're going to be looking to reduce cytotoxicity and certain systemic toxicities. So a lot of that's going to translate, and we wouldn't actually have to pivot anything. As far as moving to another indication, we would need to do a bit more PK. So we have done a little bit of PK, but we don't know exactly where the ACEs accumulate. So if we're looking at something like a UTI drug or something like that, we don't know necessarily how much of the ACEs get to the urinary tract. So I think the activity is not really a question for us, because we do have broad spectrum activity against gram positives and gram negatives, and it doesn't seem to matter if they have any sort of resistance elements. So it's really more about the in vivo performance of the compounds. And like I said, we actually transitioned from some of those other indications to NTM. So we do have a little bit of work in other infections.
Great.
Thanks.
Time's up.
Sounds good. All right. Well, thank you, Robert and Alex, very much for your presentation.
Thank you.
Thank you.
Moving on to the next company, Palm Therapeutics.
At Palm Therapeutics, we're developing next generation therapeutics to treat cancer at the root. Cancer is a global health crisis which affects 17 million people each year and results in 9.6 million deaths. These staggering numbers make it abundantly clear that we need better cancer treatments. At Palm, we're focused on a process called palmitoylation, which mediates cancer progression in 40% of all cancers. We've developed an innovative class of new drugs called DPALMs, which shut down this palmitoylation process and selectively kill cancer cells. We were recently awarded a $400,000 SBIR from the National Cancer Institute to apply these to the treatment of NRAS mutant melanoma. This is the most aggressive form of melanoma and occurs in 20% of all patients. The global cancer therapeutics market is an estimated $128 billion a year, with our initial indication in NRAS mutant melanoma having an estimated market of $530 million a year. Using our approach, we're filling an unmet need in Ras-targeted therapies. This is a huge area right now with several companies focused on KRAS and HRAS mutant cancers. However, no one is focused on NRAS mutant cancers, which actually make up 8% of all cancers. And that's exactly the need that Palm Therapeutics meets. Our current status is that we've identified hit compounds, validated the underlying biological mechanism of these drugs, and done a successful in vivo proof of concept in a mouse melanoma model. A PCT for this work was filed in March of last year, and we've negotiated an exclusive license from UC San Diego. As I mentioned, we were recently awarded an SBIR and will begin this work in December of this year. Our team consists of myself, Andrew Rudd, as co-founder and CSO. I received my PhD from UC San Diego, and I'm the co-inventor of the DPALM technology. Our scientific co-founder is Professor Neil Devaraj, a professor of chemistry at UC San Diego. Additionally, we're advised by a series of experts in drug development, medicinal chemistry, and oncology. Our current plan is to, in the first year and a half, engage in the optimization of our DPALMs to improve selectivity and potency towards NRAS. We'll then take our best performing compounds forward to in vivo preclinical studies in order to identify a lead compound for IND enabling studies. It's during this point that we'll explore strategic partnering, licensing, and acquisition deals. Again, my name is Andrew Rudd, and thank you for your attention. I encourage you to connect with us via email or social media. I've also set up a Calendly for one-on-one meetings today if anyone's interested. Thank you.
Thank you, Andrew. Judges, would anybody like to go first?
This is Gladys.
Hi, Gladys.
Hey, I have a question. Can you tell me a little bit about the mouse studies and the dosing? Are you, doing IV at this point? Can you just expand on that?
Sure.
And what doses you explored?
Sure, yeah. So really it was just a preliminary proof of concept. So we did a 20 mg per kg IP dosing. And we saw a significant reduction in the rate of tumor growth. So we haven't flatlined tumor growth, but that was just our first generation compound. So since that time, we've actually discovered more potent and selective molecules, and that's also the biggest aim of our phase 1 SBIR is to improve the potency and selectivity of our compounds.
Okay. How long was it? Was it like 14 days, seven days?
We did a 7 day. Yeah.
Okay.
Once a day dosing.
Okay. Yeah, that's a low amount. And did you take a look at the tissue afterwards or anything?
Yeah, so at that dose, we don't see any signs of, tumor tissue or healthy tissue?
Both.
Okay.
Which one did you do?
Sure. Well, yeah. So just looking at the site of injection and just systemically, we don't see any obvious signs of organ toxicity or anything like that. And then doing histology on the tumor, we see that we are able to induce apoptosis in these cancer cells, and that we shut down cancer signaling pathways as well. So looking at phospho-ERK, we see that we're shutting down phospho-ERK activation.
Okay. Oh, that's good. That's good work. Thank you.
Either Alice?
I was curious about the selectivity. So you'd mentioned that this is like for the NRAS. So I was wondering if you mentioned that you guys have looked into the mechanism. Is that something you can elaborate on?
Yeah, sure. Yeah, so really the mechanism of these molecules is they're targeting this post-translational modification called palmitoylation. And so this is a modification on cysteines of certain proteins in the cell. So basically palmitic acid is attached via a thioester linkage. And so what we're doing is we've actually designed small molecules which engage in a cleavage reaction with that thioester bond and remove it. And why that's interesting for NRAS is that it's well understood that for NRAS, palmitoylation is required for its signaling activity. When you remove that or you mutate out that cysteine, you completely shut down downstream signaling activity. And so that's what we're going after. In terms of the selectivity, what we're doing is we're designing these small molecules to engage with favorable interactions with basically the adjacent amino acid residues, as well as what we kind of envision as this selective pocket, which is the interface between this palmitoylated protein and the phospholipid membrane. And so currently that's the model for this sort of pseudo drug pocket that we're targeting.
The pocket in NRAS or in the?
Sorry, so we're targeting the palmitylation site of NRAS.
Yeah. Okay.
And so basically, we think of that as a druggable pocket. Mm-hmm.
Okay. Thanks.
Alice Chang?
Sure. So in your data, I think I just saw your drug compared to just a vehicle. Did you do any testing with any other controls?
Yeah. So no, we haven't done any other mouse experiments, but that's also part of our planned work going forward is trying to do more extensive efficacy studies in a mouse model. Absolutely. We're also interested as well in exploring potential combination therapies, which yeah.
And then I think there was another company also targeting melanoma. So I'm just wondering how you guys plan to recruit for clinical trials, or how you're thinking about the patient population.
Yeah, so that's something that I'm not an expert in, but looking ahead, so certainly we'd be, as my understanding, we're at the point now where we can actually do clinical trials where we're enrolling people with an NRAS mutation. So I think that's where this is going to be most beneficial, obviously, because it's an NRAS targeted therapy. And along those lines, it also gives us the opportunity to target other cancers with high rates of NRAS mutation as well. So melanoma is our first indication, but there's several other cancers, as I mentioned. 8% of all cancers actually have an NRAS, are driven by an NRAS mutation. So there's a lot of potential out there.
When you say 8%, is it 8% of like a variety, or is it just like of the types of cancers?
Basically, it's 8% of basically all cancer patients have a NRAS mutation.
Okay, so but for any given cancer, it is a low percentage.
It's a variation, yeah. So melanoma has a relatively high mutation rate. So 20% of melanomas have an NRAS mutation.
Would that be considered the high end?
That's on the high end. Mm-hmm.
Okay.
Great. Sounds like some, from an IP standpoint, some patient selection strategies you could implement as well by having a companion diagnostic potentially.
Potentially, yeah. Again,
you thought about that?
It was something I thought about a little bit, yeah. But again, not something I'm too familiar with at this point.
Sure. Okay, great. Well, I think we're right at 12 minutes. So, Andrew, thank you so much for a great presentation.
Thank you.
And next up, we have Visicell Medical.
We are Visicell, GBS for cell therapy. My name is Mia Du, founder and CEO of Visicell. I have more than 15 years of directly related experience and have previously contributed to successful exits of 2 biotech startups. Immune cell therapy saves patients like Emily, and the market is exploding. However, a major challenge in the space is we currently do not know whether these cancer-directed cells are hitting their marks or not as intended in the patients for a critical, timely clinical decision on treatment strategy or change the course. Clinical attributes take too long and is life or death for the patients. Visicell has the solution, and our patent-pending solution labels the cells with biodegradable nanoparticles before injected to the patients and uses standard MRI available in most clinics to monitor proper tumor engagement by these killer cells. The threshold market for immediate revenue generation is tremendous at $1 billion, but we don't stop here. This is our market entry tactics to further reach into the clinical space where there are more than 1.8 million new cancer patients every year. Our business model is a direct B2B sale of product kits and augmented services to immune cell therapy developers in the industry. Our go-to-market strategy is to enter at the preclinical development and become embedded in our customers' product as they advance to the clinical space to treat cancer as a companion therapeutic. Unlike our competitors, Visicell provide real-time cell location information at 20 times cheaper and 5 times faster using conventional MRI equipment available in clinic for early outcome assessment. We have a significant market traction with 276,000 in sales, shortened and clear FDA path, and 3 partnerships using our product. We have a great team with more than 40 years of directly related experience and accomplished advisory board of serial entrepreneurs and stakeholders in the industry guiding our strategy. We are raising $1 million in convertible notes and have raised $500,000 to date. We plan to use the fund to take the company from our current traction to further validate the business model and develop preclinical evidence as imaging-based biomarker. There are a lot of activities in the space with CRO and cell therapy companies like Novartis paying an upward of $500 million for companion diagnostics or imaging biomarker. Together, let's put GPS cell tracker in every cell therapy. Thank you.
Comments or questions?
Well, I think the fact that you have sales is something that typical kind of like early stage life science companies don't have. So congratulations on that. But I was curious, based on those sales, like what have you learned? Like who are you selling to, and what have you learned from these initial conversations that will inform your go-to-market?
Yeah, so initially we started with service contract sale, depending on the stage of their cell therapy product development. So between about $150,000 to about $300,000 per contract. And that is, we started with our academic collaborator who turned into our customer. And then we currently talking to the cell therapy companies, 3 cell therapy companies. What we learned is service contract sale takes a long time. It's the sales cycle is about 9 months to 12 months, and that is before COVID-19 period. So we had 4 sales pending when we were able to close. 3 were still pending when COVID-19 hit. So it necessitates our business model to pivot or adapt into the COVID-19 situation, which how we responded it was that we did the product launch and we started selling the product kit. So we were able to sell 25 kits in the past about since April. So about 6 months now. Yeah, 6 months. And then that actually kind of shortened the sales cycle. And we are currently basically validating that new business model of product kit sale with COVID-19 special of augmented services add to it.
So are these kit sales to academic researchers or to early stage cell therapy companies?
To academic researcher and well cell therapy company.
Okay, so I think that that's one thing that I've noticed when folks create tools, they create for researchers first, but that transition into the industry is a pretty wide gap in terms of trying to understand. And yeah, and a lot of the time they want the service because they don't want to deal with it. So yeah, I think that more conversations will help to understand.
Yeah, you are absolutely right. Our conversation with medium-sized and big cell therapy companies do tell us that we want end-to-end solution where we hire you, pay you, and then you give us beautiful images to include in the, IND filing or to talk to the physician. So you are absolutely right. So we have a package options creating that, although we are adapting to the COVID-19 situation at the moment.
Good luck. Thank you.
Thank you, Alex.
Can you also talk a little bit about, I guess, piggybacking off of that, in terms of training and when you're selling your kit, what other resources do you need to provide for them to just use off the shelf?
Off the shelf? Yeah, so we made our product kit to be as easy as possible. So it is a simple SOP that comes in the kit, but we do provide customer service, especially right now with COVID-19 special. We are adding the initial analysis of in vitro analysis of our product on their cell therapy product for free. So like viability study and labeling efficiency study, we basically added on as COVID-19 special. But to answer your question, the bigger part is where, MRI sequences that work on these nanoparticle and MRI sequences and analytics that give us the informative data on how the cell therapy is performing or targeting, that we have a product package that we have collaboration that we are developing together, as well as the product package option that we are working on. Did I answer your question?
Yes, thank you. And then one more in terms of just, can you share any data on how this affects just like cell viability, if, there's the degradation profile, anything on the tech side?
Sure. Can you see the slide now, or should I share it again? I think I need to share it again.
See it.
You can see it now.
Let me see. Can you see the slide? So
Slide was on and then it's out, not there anymore.
Okay. Let me see. It was a technical problem with the.
There you go.
So let me see. So we can start with this. So the underlying core technology was published in Nature Medicine as myself as the first author. And can you see it?
Mm-hmm.
Can you see the slides? Okay.
Yeah.
So in terms of how the nanoparticles, this is how the nanoparticles look, and they are internalized inside the label cells, as you could see it in the TEM of the images. To answer your question about the viability and the toxicity, we have found no significant toxicity in the label cell product as we look at it by panel A, which is the apoptotic assay, and panel B, which are looking at the metabolic activities with the MTS mitochondrial assay, and C with the redacted oxidative species. So we see no difference between labeled and non-labeled cells. And what I'm not showing here is that we also look at the differentiation potential and the functionality of those therapeutic cells, and it has no effect in 7 different types of cells that we tested.
So Maya, yeah, this is Gladys. While you have the slide up, is it hi, nice presentation. Are you giving it with the cells at the same time? To give it time to tag and be able to track the cells, like you said, so it's kind of dosed together with the cell therapy?
Yeah, so our current protocol is we label the cell product in the laboratory before giving it to the patient.
Okay.
So that's the product that we have fully developed and tested and validated in preclinical model as well as in one phase 1 trial. In terms, let me see, I think you're asking if we give the nanoparticle directly to the patient to detect the cell product in the patient.
Right.
Yeah, so our current product is not tailored towards that. We have a early development done in-house, but that's not a fully developed product yet.
Okay. And then just on this slide, it looks like it's stable for a few days because you're going out 4 days here.
Yes.
It looks like. Okay.
Yes.
Just on its own, what's the clearance? Like if you just give it just to see how the clearance works, what's the typical average time to clear?
It is about 14 days. It goes through the regular reticular IES system. And we do have the new product that are patent pending that we are taking it to the clinical grade product. So for that, we haven't done the study yet.
Okay. Well, thank you.
Thank you, Gladys. Either Alice, any further follow-up beyond your questions earlier? Okay, well, thank you so much, Maya. Great presentation.
Thank you.
And why don't we turn now to, we're actually staying on schedule here for everybody that planned accordingly. So why don't we turn to Cellaccura Biosciences.
First of all, thank you for the opportunity to present. My name is Helen Poltsam, and I am the CEO and co-founder of Cellaccura Biosciences. At Cellaccura, we develop precision gene therapies for the brain. So what does it mean and why is it important? Although most neurological disorders affect certain cell populations in the brain, contemporary therapies rarely do. For example, opioid pain medications effectively engage analgesic system, but they also suppress respiration and cause dependence through completely different targets in the brain. So in short, although efficient, they lack specificity to the desired target. Thus, the key question is how to restrict therapeutic activity to desired targets. What makes designing therapies even more challenging is that oftentimes targeted cells and side effect causing cells are physically intermingled and anatomically co-localized. But notably, they differ by genetic makeup, and this creates a huge untapped opportunity for therapeutic design. At Cellaccura, we've developed a patent-pending cellular engineering platform called CellSelect that allows us to gain selective control over any cell type in the brain by harnessing the genetic programs that define the cell. This technology enables us to kill, activate, or inhibit any cell type in the brain. And with the technology, we're aiming to create one-time curative therapies for neurological disorders that were unaddressable so far. So our first two focus areas are epilepsy and chronic pain, but more importantly, with an exponentially growing understanding which cell types affect which neurological disorders, there's a rapidly growing market opportunity for this technology. In terms of where we stand today, we've completed our in vitro and in vivo proofs of concept, and we've raised $185,000 in non-dilutive funding. With additional non-dilutive funding that we're pursuing right now, we're aiming to deploy the technology to animal models of chronic pain and epilepsy. Last but not least, let's talk about our team. My prior experience includes investment banking, as well as co-founding a company that turned into a leading B2B accelerator in the Bay Area called the Alchemist Accelerator. The inventor of the technology, Dr. Alan Herman Poole, is a neuroscience postdoc at Caltech, and we're advised by Dr. Yuki Oka, a professor of biology at Caltech, and the technology was developed at his lab. And right now we're looking for collaborators for our epilepsy proof of concept. With that, I would like to close. Thank you, and I will take any questions.
Great, judges. Anyone would like to go first?
Yeah, I'm curious about, you had mentioned that you guys have done in vitro and in vivo proof of concept. Can you explain what that was, like in terms of the findings, and then how does that, are you basically doing the same but in a different mouse model, or in these like the kind of indication mouse models that you're going to take a slightly different approach?
Yeah, so I'm joined by my co-founder, Dr. Herman Poole, and I think he's going to elaborate on that.
Yes, yeah. Let me see if I can I change the slides? I see. So essentially, I'll say a few words what the competing technologies are, and then explain what's our innovation. And essentially, for any kind of precision brain therapies that rely on gene therapy, there's a few different modalities that people have pursued in hopes of achieving kind of specific and targeted therapies in the brain. One of which, where there's actually quite a bit of an innovation in Southern California as well, is engineering the capsid of gene therapy vectors such as adeno-associated viruses to be specific for cell types that you're targeting. And that works beautifully for to differentiate cells that are very different, such as neurons from glia and so forth, but which lacks the precision to differentiate between different types of neurons. The other approach that people have taken is to use promoter driver viruses, where the specificity is achieved by virtue of a short promoter sequence that hopefully will target the expression of the kind of the effector gene to the desired cell population. And that works beautifully, but only maybe like for like 1% of cases that you would design them for. And that's by virtue of the fact that the DNA sequence that should ensure the cell type specificity usually does not fit into kind of contemporary viral vectors. And the approach that we're taking is using in vivo gene editing. So essentially using a method that will probably be double Dutch for most of the audience, but using a method called homology-independent targeted integration. That essentially enables you to do high efficiency knock-ins to genomic loci that are active in a cell type specific manner, okay? And so instead of just basically trying to fit as much stuff into a viral vector as possible, what you can do is essentially knock in your therapeutic gene that either silences, kills, or activates the cell to the genomic locus that is expressed in a cell type specific manner. And that essentially only will guarantee the expression of the therapeutic agent in the cells that you go after. And so we've done both the in vitro as well as in vivo proofs of principle. So for in vitro proofs of principle, we're able to achieve efficiencies that go to 80% or above. And also we've implemented a system with AAV viral vectors, getting robust efficiencies going up to 50% of targeted cells in vivo. And so that's kind of what we're using as, or the innovation essentially is we figured out how to do high efficiency in vivo knock-ins using AAV viruses.
Great. Thank you very much. Alice, or yeah, go ahead.
Can you talk a little bit about, I don't actually know which gene you're targeting, but in the brain, at least when I think about something like depression or one of the broader indications that you mentioned, it's like dopamine, are there just compensation mechanisms that would make up for it if you silence it in one cell? And or when you talk about pain, do you actually want to 100% get rid of whatever you're trying not to express?
Yes. So like, first of all, I think like the question I should probably answer is what kind of cell types are we targeting? And the two indications that we're going after are focal epilepsies, and the other one is kind of neuropathic pain that we're targeting. And so for each condition that we can target, we in fact have to have the cell population that we're going after. So I'm going to bring an example for pain and then for epilepsy. So for pain, what has come out of our preliminary work is what we found is a cell type in the brainstem that mediates opiate analgesia. So this is the neat technologies, like what you can do currently is essentially find if you know which brain structure controls the kind of the therapeutic effect using technologies such as kind of RNA-seq-based stimulus to cell type mapping, you can identify the cellular substrate that mediates the effect. And so essentially we have both the cell type as well as the genes that we're targeting to get the cell type specific expression of a chemogenetic activator to essentially just induce analgesia with a therapeutic that the rest of the body does not see. So we have the target, and in preclinical work, we're currently deploying this system for selective activation of this opiate analgesia mediating cell type in the brainstem. And on the flip side of the coin, for epilepsy, it's much more crude. In epilepsy, we're targeting just the excitatory cells by virtue of targeting genes that are expressed in glutamatergic neurons in the temporal lobe, and then using this as a way to gain selective control only of these excitatory seizure-causing cells.
So with that said, this is Gladys. Are you stratifying within the patient population? So for example, the epilepsy foundations and, they have kind of a breakdown of the different genetic mechanisms around epilepsy, right? So that you can maybe focus on a certain genetic type to begin with. And then there's obviously those that just kind of are geared towards pediatrics. So I'm assuming that as you develop it, you'll partner with organizations like this to kind of stratify within the patient population, right? So that you can really target the treatment.
So yeah, that's a very good point. So essentially, we've spent quite a bit of time to figure out what is actually like a realistic clinical cohort with that we can target. And the approach that we are essentially going after are kind of rare epileptic disorders that are focal, for which there is no kind of robust pharmacological treatment out there. So tuberous sclerosis complex was one of them that we would like to design our first therapy for, and then provided this is efficient, we could like essentially go from there. But essentially go
through off-label, prescription later to sort of expand our market.
But essentially, to go, like our current plan is to go after rare diseases for which that would be kind of for which there is no kind of clinical clinically efficient treatment to go from there.
Okay, thank you.
All right. Thank you, Helen, so much, and Cellaccura for a great presentation. Now, I believe, Mark, are you with Alyra? Are you on?
Well, thank you. Thank you, everyone, for your time, and thanks to the organizers for the opportunity. I'm Mark Benedict. I'm CEO and co-founder of Alyra. I have a PhD in genetics, but I've been in business development in biotech my whole career for about 25 years, working at Pfizer, Elon, Vertex, other venture-backed companies as well. And I'm going to talk a little bit about Alyra today. We were founded in 2020, and just a few months ago, we are focused on melanoma, stomach cancer, and colon cancer, but melanoma is our primary indication. And we've developed ALY-101, which we have rights to from UC Irvine and the Italian Institute of Technology in Genoa, Italy. Okay, slides are too small. So
no, I think that you're seeing the presentation mode, and we're actually seeing the presenter mode right now.
Okay, let me fix this. Let's see. Okay. How is this?
We don't see anything.
How's this?
That's better.
Good.
Can everyone see it?
Yeah, we see your first slide.
Okay. Okay. So let's see what's next here. And now I can't move. Here we go. We are. Let me catch up from where I was. We're raising a convert, a 250K convert to expand on the in vivo proof of concept data we have in melanoma, and that's really a bridge to a Series A, where upon close of that round, we'll be probably about 12 to a little less than 12 months to an IND on our clinical lead. The market for this is actually quite large. There's been an attempt in the past in pharma to develop PAC1 inhibitors. They've approached it through a traditional ligand receptor approach, antagonist receptor approach. We actually have an allosteric inhibitor, and there's been a lot of work at Genentech, Novartis, BMS, and other companies looking at this space. We see a lot of, we're going to piggyback off of that. We see a lot of potential to develop combination therapies with other targeted therapies, really focused on melanoma to start with, because in stage 3 melanoma, there's really not a lot of good therapy available without serious adverse events to patients that provides durable responses. We see a $3.1 billion TAM for this product, with a very, very conservative price point of $10,000 per patient per year. Just to give you a sense of where that stands, targeted therapies in the US are about $150,000 per year per patient. So our focus really, again, is on patient outcomes. And as I said, about $150,000 per year per drug per patient, with poor response rates, which I'll talk about in a minute, which are accompanied by very severe side effects. Those include serious adverse events like grade 3 fevers, 103 and above, disabling rashes, neuropathies, and other problems. And a big problem with the new therapies is really chemo resistance. So for instance, in melanoma, there are a couple of new products called BRAF inhibitors, which really develop a resistance, patients develop resistance to quite quickly. We're going to leverage our experience to treat this early stage product like with a pharma development focus. A team and myself, we have about 75 years total experience in drug development. We have a pharma array system where we're planning on screening synergistic combinations with existing therapies like Tykerb, Visicell, Zelboraf, other products to look for combination synergies, which we've already seen in the literature from Genentech and Novartis. And we're going to expand on that data to identify potent and safe drug combinations with superior therapeutic indices. Where's the area today? Well, really the targeted therapies have about a 45% success rate, anywhere from 18 to 45% for the whole category in stage 3 melanoma. But 45% success rate can translate into 1 month delayed tumor progression with disabling side effects. So we really want to improve the patient outcome and synergize with these therapies, existing marketed therapies, so we don't have to use the same amount of drug to get a better effect. Our product is very specific for PAC1, and we've de-risked the program quite a bit. We have a backup compound to our lead.
I hate to break in here, Mark, but I have to follow the rules, and we've gone way past time. I apologize.
Okay,
so
sorry, I couldn't see the timer with the full screen, so I'm a little lost.
Yeah, well, I'm sure you can get some of that in answers to the questions, Mark.
Sure.
So why don't we turn over to the judges?
Yep, great job.
Thanks, Darren.
Yeah.
So very quickly, Mark, beyond melanoma, what is the strategy?
So beyond melanoma, we have a biomarker strategy we're pursuing, and we're getting some proteomics work being done at MD Anderson. PAC1 is present in quite a few tumors at high amounts. We're focused on GIST and serrated colon cancer, for which there's really very little available. And we've been talking to people at Irvine about a strategy for GIST, for gastrointestinal stromal tumors. And then outside of those 3 solid tumors, which is about 85% of the PAC1 expressing tumor populations, there are a number of hematological malignancies. Diffuse large B-cell lymphoma and MDS are 2 possibilities in that space.
Thank you.
I think you also mentioned there were some rare disease indications. Can you talk about some of those?
Yeah, so the PAC1 inhibitors were originally designed as Fragile X syndrome drugs, and Fraxa, a company here in town in San Diego, which was bought for, I think, about $180 million several years ago, was focused on that area. That was the unsuccessful traditional receptor antagonist approach. Those compounds never really made it to the clinic. So I have ties to the Fragile X Research Association. The other areas where we want to pursue, where I have relationships in Texas, are Ewing sarcoma, and which is a rare childhood diffuse cancer, solid tumor, and also neurofibromatosis, where I also have ties to the patient advocacy groups there. I advise a rare disease group, a nonprofit here in Encinitas, so I've got a lot of connections in those places.
Awesome. And then one quick one. I'm a little bit confused. Are you guys in San Diego, or is it a distributed team?
It's distributed. So I'm in San Diego along with Alessandro Monge, who is a classmate from Rockefeller, where we did our PhDs. We're both here in San Diego. Anand Ganesan is at UC Irvine. He's the co-inventor on the patent we've got rights to, along with Marco De Vivo, who is at the Italian Institute of Technology. And then Jay Studemeyer is our non-clinical advisor. He comes from a long tenure at Genentech and Chugai. He's currently consulting for Deerfield for 3DC. And he is in Austin, Texas. I have a lot of relationships in Austin. I'm consulting with an advisory group at a secondary fund here in Encinitas. We manage a portfolio of life science companies in Texas.
Mark, you had mentioned that this could be used in combination. So I was wondering, as you move forward, is like co-development kind of more in the strategy, or are you looking to?
You mean a strategic partner early on? Yeah, so we started talking to pharma, and we actually have an ongoing discussion with a group in Germany, a pharmaceutical company. Our idea really, right now we're getting some pharmacokinetics work done, formulation work done, and then some toxicokinetics. So we'll have, I think, a very robust data package before we proceed with our IND enabling work. And that's really coming from people from my background and people at pharma we've just talked to informally at Pfizer. I used to run the Pfizer incubator here in town in San Diego, so socialize a lot with friends at Pfizer. Certainly open to that. I think we've talked to some investors informally that have come back with very different perspectives, but I think co-development with an emerging product would be great, especially since we'll have a biomarker panel probably ready to run sometime in the middle of next year.
Have you spoken with our team in San Diego, like the Alexandria Venture team? I
have not yet, actually. So I've been somewhat I'm somewhat conservative because I've seen a lot of early stage programs where I scratch my head at the valuations. I have not yet talked to anybody, and I would love to chat with you after this meeting, because we're having work done in overseas and here by vendors, and then we're going to raise that note. And people we're talking to already are saying, why not just make it a tranche instead of raising debt and then equity? So we'd love to continue the conversation.
Yeah, I'll connect, and I can connect you with our San Diego team. We manage our regions by region. So.
Oh, great! Thanks, Alice.
Yeah, no problem.
Any more questions?
Any questions from the attendees? Haven't seen any come in for a little bit. Okay, Mark. Well, thank you so much for a great presentation, and best of luck to your company.
Thanks, Matt.